Gene therapy with you today. Objectives: to explain the concept of gene therapy and its different manifestation, as well as examine the issue's possible benefits and disadvantages for world health. Introduction: the module focuses on gene therapy, a modern biotechnology product that allows the people to better understand the molecular foundation of health and disease. What is gene therapy? Gene therapy is a new strategy for treating or preventing disease that involves the use of genes. In the future, instead of utilizing drugs or surgery, doctors could use this technology to treat an illness by inserting a gene into a patient's cells. Researchers are experimenting with a variety of gene therapy procedures, including replacing a disease-causing mutant gene with a healthy copy. And ay napakaganda kung iisipin, ngunit ang gene therapy ay na isang promising therapeutic option para sa iba't ibang karamdaman ay isa pa ring mapanganib na pamamaraan na kailangan na masusing pagsasaliksik upang matiyak na pareho itong ligtas at matagumpay. Tandaan natin na ang gene therapy ay kasalukuyang iniimbestigahan lamang para sa mga sakit na walang alternatibong paggamot. There are 4 different types of genetic diseases. First is single gene changes. Single gene disorders are caused by mutation in a single gene. Single locus gene is detected and responsible for disease. The disease is 100% heritable. Single gene diseases are extremely uncommon on their own, yet they affect truly 1% of the population overall. At dahil ang mga karamdamang ito ay sanhi ng isang gene, madali silang masubaybayan. Example include sickle cell anemia, hypercholesterolemia, and cystic fibrosis. Second, multi-gene disorders. 2 or more genes are defected and responsible for the disease and is less than 100% heritable. This disease may be dependent on environmental factors and lifestyle. Example include heart disease, cancer, and diabetes. Ang mga uri ng multi-gene o multifactorial na sakit na ito ay magiging mapakahirap na gamutin ng mahusay gamit ang gene therapy. Ang pangatlo naman is mitochondrial disorders. Ang mitochondria ay gumaganap bilang pabrika ng enerhiya ng ating katawan. Ang mga sakit na mitochondrial ay pangmatagalan, namamana at madalas na hinanang mga kondisyon kung saan nabigo ang mitochondria na magbigay ng sapat na enerhiya para sa katawan upang gumana ng normal. Kaya mutation in the mitochondrial DNA causes disease that affect many organ systems, including the cell of the brain, nerves, muscles, kidneys, heart, liver, eyes, ears, and pancreas. And last, we have chromosomes abnormalities. Sometimes, complete chromosome or part of the chromosomes are missing, duplicated, or modified in some way. Birth malformation affecting the brain or other region of body are commonly caused by chromosome abnormalities. The mother chromosomes are 23 and the father chromosomes are 23 in typical fertilized egg cells. As a result of a fertilized egg usually contains 23 pairs of chromosomes. Ang terminong trisomy ay halimbawang sakit nito. Ito ay tumutukoy sa pagkakaroon ng 3 chromosomes kaysa karaniwang pares. Sa karamihan ng mga kaso, ang isang sanggol na may Down syndrome ay may 3 kopya ng chromosome. Bali ito ay mayroong bilang na 21 kaysa karaniwang pares at ang condition ay na iba pang kilala bilang trisomy 21. Kasama sa iba pang karaniwang trisomies ang trisomy 18.
Types of gene therapy. Somatic cells. Our non-reproductive somatic cell therapy is viewed as a more conservative, safer approach because it affects only the targeted cells in the patients and is not passed on to the future generations. Ang somatic cells po ay hindi po reproductive. Ibig sabihin po, anuman pong uri ng cell sa ating katawan, maliban lang po sa sperm cell at egg cells na mayroon po, ang mga somatic cell diploid po, ibig sabihin naglalaman ng 2 set ng chromosomes. Ang isa ay minamana mula sa bawat magulang at yung isa naman po ay yung mutations ng mga somatic cell na maaring nakakaapekto po sa individual ngunit hindi naman po naipapasa sa mga supling. Ang somatic gene therapy ay angkop po nakatanggap-tanggap para sa mga maraming karamdaman. Kabilang na po dito ay ang cystic fibrosis, muscular atrophy, cancer at ilang mga nakakahawang sakit. Germline cells results in permanent changes that are passed down to subsequent generations. If done early in embryologic development, such as during pre-implantation, diagnosis, and vital fertilization, the gene transfer could also occur in all cells of the developing embryo. Sa germ cells, ito naman po yung nagre-resulta sa mga permanenteng pagbabago. Kung sa somatic cells po ay hindi po naipapasa, germline cells naman po ay pinapasa sa mga susunod pa na henerasyon. Ito din po ay kapag yung DNA ay inililipat po sa mga cells na gumagawa po ng mga reproductive cells sa mga egg cells, sperm cells sa katawan, yung ganitong uri po ng therapy ay nagbibigay daan po para po sa pagwawasto ng mga variant po ng gene which is hindi po karaniwan kaysa po sa normal na tao na nagdudulot po ng sakit na tiyak po na ipapasa po mula sa henerasyon hanggang sa ang germline therapy din po ay nagbibigay daan upang iwasto po yung mutation na mayroon po ang isang tao na nagdudulot po ng sakit na tiyak po na naipapasa sa mga supling sa susunod na henerasyon na kailangan pong maiwasan para po sa next generation ay hindi na po sila makaramdam ng pagdurusa mula po sa sakit na ito. The use of vectors. Certain viruses are used of vectors because they can deliver the material by infecting the cells. Yung sa vector naman, ito po yung mga materials na itinuturok bago po magbigay ng intravenously sa pamamagitan po ng IV. Ito po ay direkta sa isang particular na tissue po ng ating katawan na kung saan po ito po ay kinukuha ng mga individual po na tissue. Ang ilan po kasi sa mga viruses po na ginagamit bilang mga vectors dahil maaari pong ihatid po yung material sa pamamagitan po ng pag-infect sa cell. Dahil ang mga vector system po na ito ay may bawat aplikasyon kung saan po ito naaangkop. Somatic gene therapy can be broadly split into two categories. The first one is ex vivo, which means exterior, where cells are modified outside the body and transplanted back in again. Involves the genetic modification of cells outside of the body to produce therapeutic factors and their subsequent transplantation. Back into patients, various cell types can be genetically engineered. Sa ex vivo naman po therapy, ito po yung pag-alis po ng mga cell mula po sa katawan ng isang tao. Dito po ay binabago at naglalagay din po ng cell pabalik po sa ating katawan. Ang ex vivo therapy po ay kadalasang ginagamit po sa may mga sakit na nauugnay po sa dugo. Kabilang na dito yung particular na sakit po na cancer, leukemia at lymphoma at mga iba pang genetic disorder na in vivo, which means the interior, where our genes are changed in cells still in the body. This form of gene therapy is called in vivo because the gene is transferred to cells inside the patient's body. Sa in vivo, ito po yung mga bagong genes na direktang ipinapasok po sa katawan. And it's also thought to be preferred when the target is also internal organ, gaya na lang po na may sakit po sa puso. Gamit po yung in vivo gene therapy, yung mga target cells and tissue po ay kailangan po na madaling ma-access para po sa pag-injection ng mga genetic material. Halimbawa na lang po sa pag-injection ng genetic material ay yung sakit po sa mata, which is the Luxturna. Ito po ay direktang ini-injection sa mata po sa ilalim ng retina. So, in the end of my report, the bottom line po ng aking report, ang gene therapy ay maaaring ibigay po sa pamamagitan lamang ng ex vivo at in vivo ng mga paraan sa paghahatid lamang.
Viruses uses as vector. May tatlo po itong nilalaman. The adeno-associated virus, the adenovirus, and the lentivirus. The adeno-associated virus is a small, non-developed virus that packages a linear, single-stranded DNA genome. Adeno-associated viruses are small viruses that infect humans and some other primate species. AAV also may be transmitted through direct contact with an infected individual or through indirect contact with the contaminated environment. The adenovirus. The adenovirus is a non-development virus that contains a linear, double-stranded DNA genome. The ADV or adenovirus vectors constitute very powerful vehicle for the gene transfer with application in vaccination, like cancer treatment and many monogenic and acquired disease. Isa po siya sa mga nagsisilbing gamot sa mga viruses like COVID-19 po. The lentivirus. The lentivirus is an enveloped retrovirus containing a positive sense single-stranded RNA genome. The lentivirus have been used to introduce auxins into the brains of mice, rats, and primates. Lentiviral vectors are also a vehicle for gene delivery that were originally derived from the human immunodeficiency virus or HIV. Non-viral DNA carriers. Non-viral delivery methods may be safer than viral methods. Some of these non-viral methods include cationic liposomes and naked plasmid DNA injection. Cationic liposomes are positively charged lipids that can interact with negative charged DNA, forming a lipid DNA complex. The cationic liposome is one of the most commonly used non-viral gene delivery vectors, and it is widely used as standard gene vector, especially for in vitro transfection. Naked plasmid DNA injection. A gene construct is made composed of a gene of interest with a promoter to direct protein synthesis. Naked DNA refers to DNA that is not associated with proteins, lipids, or any other molecule na tumutulong para protektahan ito. An example of gene therapy clinical
That from gene therapy, Jesse Jellick Singer, it was known that Jesse had a metabolic disorder calling OTCD that causing ammonia buildup to deadly levels in blood. Jesse undergoes numerous medication and clinical trial for hypothetically treatment to his diseases throughout the developed peaks for OTC gene that produce enzyme prevent the pneumonia buildup from study to University of Pennsylvania. However, the critical trial of gene therapy was not a success because it rapid respond, causing him to having multiple organ failures and blood clotting disorder, putting him into brain dead within 4 days. And other deaths related to gene therapy includes 2 boys who undergoes treatment for real muscular diseases. Therein, after receiving high dosage, they develop liver problems that equivalent progress to sepsis. From this observation, researchers finding out the high dosage of gene therapy can cause dangerous liver toxicity, but related is from the researcher run by Astel. Therein, 4 boys after age of 5 die in critical trial after developing liver calcification. Next, the success from gene therapy. There are studies that the University of Pennsylvania having successful gene therapy swap to prevent vision loss. The LCA is a human disease that prevents the retina from react or sending signal to the brain. Then, next, the training the immune system of fighting cancer. Humoral anti-aging repertory or known as CAR-T cells therapy works by programming a patient immune cells to recognize and target cells with cancerous mutation. This gene therapy focusing on the patient T cells. Third, the precision editing for blood disorders. Throughout the method of CRISPR case, the DNA of sequence, bacterial, and crisping associated enzyme is using to editing the regularity illness. The disease is called spinal muscular atrophy or SMA is a neurodegenerative diseases that is a aging mutation that cause to grenade to kill motor neurons throughout a solution and technical issues surrounding to us whether the use for this technology is good or bad for our society. Second, the decision that people or researchers decide about which trials to normal and which are causing disability or diseases. Third, it costs which only makes it available to reach what it is popularity, society norm of acceptance towards one from. And lastly, is a technical used to modify trials for personal gain. To simply summarize our topic, overall, the gene therapy seems to modify or manipulate the impression of gene or the metabolic properties of living cells for use. The gene therapy may pose particle medicine being for humans into critical consideration should always be addressed in order for society to utilize the potentials of gene therapy appropriately since its products are being studied to treat diseases include cancer, genetic diseases, and infection diseases.